When we talk about the synthesis of proline, it's first important to understand our starting material. Our starting material here is glutamate. But where does glutamate originate from? Glutamate is formed from alpha-ketoglutarate. Alpha-ketoglutarate is, for example, it's found in the TCA cycle. And when that alpha-ketoglutarate gains an amino group, it becomes glutamate. Now, so here we're going to start off with glutamate, and this is the structure of glutamate. It's an amino acid. So we're going from one amino acid and we're trying to make a different amino acid, in this case proline. So remember, glutamate, it has this alpha carbon, the carboxyl group, the amino group, and that R group. In this case, we have 3 carbons and that carboxylate group at the end. In the first step, we want to phosphorylate. We want to phosphorylate this oxygen right over here. So we take the terminal phosphate on ATP and we attach it to glutamate to yield gamma-glutamyl phosphate. And this reaction is catalyzed by glutamate kinase. It's glutamate because that's what our substrate is, and kinase because whenever we think of a kinase enzyme, a kinase is going to be utilizing ATP to phosphorylate. So the first step is pretty straightforward. We want to phosphorylate this glutamate to get gamma-glutamyl phosphate. Now this gamma-glutamyl phosphate is now going to be reduced. So essentially, the phosphate that we just added is going to be lost. So this phosphate is going to be lost, and we're going to yield an aldehyde. So we go from gamma-glutamyl phosphate to glutamate gamma semialdehyde. And in order to reduce the gamma-glutamyl phosphate, we use a reducing equivalent, which is NADPH. Now, when NADPH is going to get oxidized into NAD+, and we lose that phosphate, and this reaction was catalyzed by gamma-glutamyl phosphate reductase. Gamma-glutamyl phosphate, it's in the name, and then reductase because we are reducing the gamma-glutamyl phosphate. So essentially the whole purpose of this phosphorylation and reduction was to get rid of this oxygen right over here. And now we have a hydrogen in that position. Now, the next step is a non-enzymatic step, and water is going to be released, and we're actually going to see that this amino group over here is going to act as a nucleophile, and it's going to attack this carbon, which is going to act as an electrophile. So we were basically setting up this carbon to become a good electrophile. So what we did was we phosphorylated, then we removed the phosphate, and we reduced it, and then now we have an electrophile, and this amino group, the nitrogen, is going to nucleophilically attack that carbon, and we're going to see cyclization happening here. But first, we must lose H2O, where that H2O, essentially we want to lose this oxygen, and we want to lose the hydrogens in this process. So right over here, the nitrogen is going to lose the hydrogens as well. So when this happens, we get this cyclic structure, and initially we have a double bond between this nitrogen and this carbon. But let's first orient ourselves so we can understand how the cyclization occurred. So when this nitrogen forms a bond with this carbon, they're going to come together, and as a result, this carboxylate group, it's turning this way with the nitrogen. And it's turning this way, and we can actually see it over here. So essentially, this carbon right over here is this carbon right over here. And it has that carboxyl group, it has that nitrogen group. So this is our alpha carbon. And this nitrogen has formed a bond with this carbon. So this carbon correlates to this carbon. And we see this double bond formation. But this doesn't look like proline yet. This is actually called pyrroline 5 carboxylate. So now we have to convert it to proline. Well, the only difference between pyrroline 5 carboxylate and proline is that proline does not have this double bond, so it does not have any conjugation. How can we get rid of a double bond? Well, we can reduce it. How do we reduce it? We use a electron donor, and that's NADPH once again. So we're going to use pyrrolene carboxylate reductase, another reductase enzyme, and we are going to reduce this pyrrolene 5-carboxylate, and we're going to produce proline. So overall, we used 2 NADPHs and 1 ATP to synthesize proline. We went from glutamate, we phosphorylated glutamate to get gamma-glutamyl phosphate. Gamma-glutamyl phosphate was converted into glutamate gamma semialdehyde. So essentially we reduced it. So there was first a phosphorylation, a reduction, a dehydration, a non-enzymatic dehydration, and then another reduction to yield proline. And that's essentially how you synthesize proline. And remember, it all starts from alpha-ketoglutarate. Alpha-ketoglutarate first gets converted into glutamate, and then glutamate can convert into proline.